Okay, so let's make a start. First of all, I'd like to thank you very much indeed, Sean, for the kind introduction, and I'd like to thank the organizers for the invitation to speak in this Freiburg Penn State virtual classroom seminar today. So I'm talking about bee pollinators and a different approach we have taken to tackle pesticide safety issues in bee pollinators. So there are a number of factors threatening bees, and so my talk is mainly focused on managed bee pollinators. At the end of the talk, I will talk a little bit about wild bees and also included in pollination services. So I think without doubt, one of the main factors for bee declines observed over the past years is definitely the varroa mite, which is known also to submit viruses and leading, for example, here to deformed wings, and that is the deformed wing virus. But there's lots of discussion and debate out there in the public and also in academia that, of course, also pesticides may play a major role in the observed declines in honeybees. If you look into the global sales of pesticides, 85% of the pesticides sold globally are acting on nerve and muscle function in insects. And these are highly conserved target sites among insects, and that could be a problem in itself. And so you don't wonder about the fact that 95% of the top 20 insecticides by sales are acutely toxic to bee pollinators, in particular honeybees. That said, there are diverse routes of exposure. I've taken this picture out of a nice review from Reed Johnson in Annual Review of Entomology. And so you see there are diverse routes, and that's pesticide overspray and things. I think most of these things are definitely unintended exposure at the end of the day. But anyway, so there's lots of debate out there. And so there are for sure combined effects of multiple stressors, which might act synergistically. And also in the last 5 years, I think we have also a number of publications seen that chemical mixtures are under scrutiny because they are synergistic and provide another harm to bee pollinators. That said, we decided to look into the detoxification capacities of honeybees at that time. The honeybee genome has been sequenced and published in 2006, many years ago already in Nature. And so there was a satellite paper claiming that there's a deficit of detoxification enzymes in the honeybee genome, and that this deficit may translate to less pesticide detoxification capability, which would then explain the species' unusual sensitivity to pesticide. Compared to a mosquito, the honeybee indeed has much lower number of detoxification genes. A different study conducted by Hartstone and Scott at Cornell University, when they analyzed the toxicity of 62 different insecticides towards pests and honeybees, their conclusion was that honeybees were not more sensitive than other insect species across those insecticides examined. First tier pesticide risk assessment for honeybees is normally oral tests and contact tests in order to look for the acute toxicity of insecticides or any pesticide possibly applied under field conditions. And there's an EPA classification claiming that those insecticides which have an LD50 greater than 11 microgram per bee are considered practically non-toxic. And that is something which I'd like you to remember later on. So that said, and all these reports, I decided internally to set up a project which was called Bee Toxicogenomics some 6 years ago. And I think so the major objectives of this particular project was to expand our understanding how insecticides work in bees, what make bees different from pest insects, to identify the molecular determinants of selectivity, because we know that there are many insecticides out there which doesn't seem to harm honeybees, although they have excellent potential against pest insects. The other point is, can we convert this knowledge we gain throughout this project into future products, making them more safe to be pollinators? And we, of course, also are looking for an improved science-based communication with authorities. We'd like to explain to them why certain insecticides are less toxic to bees than others. So it's always good to have a molecular understanding what's behind the observations. So again, this particular pie chart showing that nerve and muscle action is the preferred targeted physiology of many insecticides. And the major classes of insecticides and targets I've just briefly mentioned here, it's the organophosphates, which are on the market for almost 70 years. We have the pyrethroids, we have the neonicotinoids, and we have the diamides, which is the latest larger class of insecticides addressing insect granule receptors, which have been introduced to the market more than 10 years ago. Under particular debate, as you may all know, are a class of insecticides acting on the nicotinic acetylcholine receptor in the central nervous system of insects, the neonicotinoid insecticides. It's by far the largest class of insecticides in the world. And here I have to, and that was a starting point for us, and that was the case study we'd like to follow. Imidacloprid and thiacloprid. From a chemical perspective, they look very similar. If you are not a chemist, it's indeed very difficult to find out the differences between the two molecules. So if they are applied to an aphid, which is a pest treated with these insecticides to control it, there is no difference in the intrinsic toxicity against Mysis persicae, that's a green peach aphid. If a bee is treated with these two compounds, it turned out that thiacloprid is more than 2,600 times less toxic to honeybees than imidacloprid. So we initiated this bee toxicogenomics project in order to find out what are the molecular reasons for these observations, although they are both neonicotinoid insecticides. First of all, we isolated nicotinic acetylcholine receptors out of honeybee brain membranes and did some target binding studies. The binding of both compounds to its target site, the nicotinic acetylcholine receptor, is not different at all. So that they show similar binding affinity to the nicotinic acetylcholine receptor. In other words, the fact that thiacloprid is less toxic to honeybees is not explained by lower binding affinity in these type of receptors in the honeybee brain. So it's not toxicodynamic, it must be pharmacokinetic. And that's what we tested. So we applied thiacloprid as well as imidacloprid to honeybees, and then we analyzed what happened inside the honeybees. And what we observed was that the amount of thiacloprid is depleted within a very short time in the honeybee body, whereas imidacloprid concentration remains rather stable over 2 hours. Looking for the major metabolite in honeybees, it turned out that it is a hydroxylated version of thiacloprid. And this particular hydroxylation pattern is a typical oxidation reaction, which is catalyzed by so-called cytochrome P450 enzymes. So we have an enzyme family identified, which could be potentially involved in the selectivity issue between imidacloprid and thiacloprid. So then we switched from toxicology to genomics. And so just briefly, cytochrome P450, for those of you not so familiar with the biochemistry, is an enzyme superfamily involved in the oxidative metabolism of foreign compounds, including insecticides, of course. They are divided into 4 major clades: CYP2, CYP3, CYP4, and the mitochondrial clade. And the major expression sites include the fat body, the gut, and the Purkinje tubules, which is the equivalent of the human kidney, if you want. They are known to confer insecticide resistance in pests if pests are selected, continuously selected by insecticides. Here is a typical paper. So that is an insecticide, pymetrozin, which is hydroxylated by a P450, which is named CYP6CM1. It's present in whiteflies, and it confers resistance to neonicotinoid insecticides in this whitefly species. So they have strange names. In this case, it's CYP6CM1. Because of the fact that clade 3 P450s are mostly involved in pesticide detoxification and also xenobiotic detoxification, we thought it's a good starting point to just look into those P450s, here in blue for honeybees, belonging to this particular family or clade. So in fact, out of the entire genome, out of the 11,000 genes in the honeybee genome, we selected 27 genes which are of particular interest. And one of those genes we thought could be the factor hydroxylating thiacloprid to thiacloprid hydroxy derivative. In order to do this, we expressed all these genes individually in insect cells, high 5 cells, and we co-expressed also cytochrome P450 reductase, which is a very important enzyme, so that P450s works. And then we isolated microsomal membranes, that is the membranes where the P450s are expressed, and we tested each individual P450 for its ability to detoxify or hydroxylate thiacloprid. After 1.5 years of work conducted by one of my PhD students, it turned out that CYP9Q3 is a factor which determines the selectivity of thiacloprid or which is likely to determine the selectivity of thiacloprid in honeybees. This particular P450 does hardly metabolize imidacloprid. So it's a good finding, and this work has been published 2 years ago in Current Biology. That said, we also looked into the expression of this particular P450s throughout the life cycle of the honeybee. So is this detoxification ability conserved throughout the entire life cycle? By the way, we sequenced the transcriptome, or we sequenced 48 different honeybee transcriptomes, 8 different life stages, honeybees isolated or sampled from 3 different hives at 2 different time points in the year, and we came up after assembly of the raw reads with patterns like this, showing for each P450 in the honeybee genome, which is called the cypome, that you can see here. The expression level of the 2 enzymes which are supposed to be involved in thiacloprid detoxification, CYP9Q2 and CYP9Q3, you can see here. They are highly expressed in adult bees, which makes complete sense because here it needs to be detoxified. And not only, of course, insecticides, but also, of course, xenobiotics such as plant allelochemicals. We do not know what these particular P450s normally detoxify. But there is, of course, also a difference in expression level between worker bees and early larval instars. It's more than 100-fold difference, as you can see here. That said, we looked for further lines of evidence and looking into how we can further confirm by, for example, by genetic methods that these P450s are involved in thiacloprid detoxification. These are honeybees in a typical cage you use for an OECD toxicity test, and they were treated with 1 microgram of thiacloprid. They look quite healthy at this particular concentration, which is normally not acutely toxic to honeybees in this setup. Then we injected double-stranded RNA, silencing CYP9Q3 transcripts in the honeybee. And we applied 1 microgram of thiacloprid again to those bees where we silenced CYP9Q3. And as you can see here, they look quite unhealthy, and they show typical symptoms of poisoning, what you would expect after neonicotinoid treatment, at least if you take one of those which are more toxic. So here we provided a further line of evidence that CYP9Q3 is involved in the detoxification of thiacloprid. The next line of evidence was constructed by engineering transgenic flies. We used Drosophila as a model where we introduced CYP9Q3 gene, the CYP9Q2 and CYP9Q1 to be complete, into different lines of Drosophila into the genome. And then we tested these Drosophila strains for the ability to survive thiacloprid treatments. And that's what you see here. Here are the flies which are ectopically expressing the honeybee P450 CYP9Q3. They survive thiacloprid treatment at rather high concentrations, where those flies expressing, for example, 9Q1 do not survive the treatment, as well as the control flies. They are all dead. So this is a nice transgenic model which confirmed that CYP9Q3 is indeed the factor providing selectivity towards thiacloprid. So that is a transgenic bee-lysed fly line which expressed this enzyme and its resistance to thiacloprid. Looking into the tissue expression levels of CYP9Q3, that's what you see here. The major expression sites in the honeybee are the Malpighian tubules as well as the brain. And that makes complete sense because in the brain, the nicotinic acetylcholine receptors are located. And before thiacloprid is reaching those receptors, it is fairly likely to be detoxified by high levels of CYP9Q3 in the honeybee brain. And that is something we also showed later by using antibodies directed against the protein. It's not just the transcript level, but it's also the antibodies against the protein. We were able to show the presence of this enzyme. That said, neonicotinoids are, of course, still under public debate. And I have not expected, to be honest, that these particular studies would really help to understand better the risks posed by exposure to neonicotinoid insecticides. But you have to keep in mind there's ongoing work in order to optimize chemotypes addressing the nicotinic acetylcholine receptor in insects, because it is so an important target and it's addressed by many different insecticide types, not just by neonicotinoids. So, but its important message here is a take home. Neonicotinoids are not all equal in terms of honeybee selectivity. I think that has been shown here. And you have to be aware that not all the modulators of the insect nicotinic acetylcholine receptors are neonicotinoids per se, even though some people may tend to say it's all neonicotinoids because they're acting on the nicotinic acetylcholine receptor, which is, from a chemical perspective, nonsense. So in order to explain it a little more detailed, so if you do a cheminformatic analysis on chemical similarity relations between agonists of the nicotinic acetylcholine receptors used as insecticides out there in the field, you see here the neonicotinoid cluster in the middle, and then you see also other clusters out there. And two clusters are marked here. And I'd like to draw your attention to the butenolide cluster. The butenolide cluster is a different chemotype. It has a different pharmacophore function, and it's chemically different from the neonicotinoid insecticide, as well as the sulfoxamine cluster, or, for example, nicotine. Although all these compounds are classified in group 4, that is the mode of action classification scheme by the Insecticide Resistance Action Committee, they are all competitive modulators of the nicotinic acetylcholine receptor acting at the so-called orthosteric site. That is the site where acetylcholine, the natural neurotransmitter, binds to the receptors. You see different subfamilies. We have the neonicotinoid clustered here as subfamily 4A, but we have the butenolides as 4D, and we have the sulfoxamines as 4C. So they are chemically different, although it is very difficult for a non-chemist to really see this. It's very difficult to explain it also to the layman. Why are these compounds chemically different, although they look quite the same? So, and Einstein already said everything should be made as simple as possible, but not simpler. So be aware. And there's one thing I'd like to mention here, because there has been a publication in 2016 explaining to people or to the layman why are these different chemotypes, for example, in this case, fluopyradiferone and sulfoxaflor, why are these neonicotinoids? Because a very simple message. They bind to the nicotinic acetylcholine receptors. It's a yes for the neonicotinoids, it's a yes for the other two. They function in a systemic way. Yes, okay, that's true for all of them, and they are toxic to bees. Oh, yes, they are, of course, these are insecticides, and that if bees are exposed to them, they could be toxic. Depends on the way of exposure. But this is by far too simple, to be honest. If you would ask the same question in a biological context with other pictures, for example, and they say that the property clearly show they should be classified as neonicotinoids because they are the same. But if you ask the question, cats and dogs, are they bears or not? And that is something, they are all mammals, yes. They are carnivores, similar to bears, and they have 4 legs, similar to bears. So the conclusion out of this, if we follow the same rationale, is that their properties clearly show that they should be classified as bears. No one would do this, but that is the difference, I think, between biology and chemistry, but I would not go into more detail here. We used this particular, so the tool we developed, the P450 expression library, was used, and we found out that this particular chemotype, fluopyradiferone, is also metabolized and detoxified by CYP9Q2 and CYP9Q3. That's very interesting, and that's important knowledge also later on in argumentation with the authorities when we need to explain why these compounds are less toxic than neonicotinoid insecticide in acute tests. So what else can we do? And so we thought, okay, now we know the enzymes which are involved in the detoxification of certain compounds, such as, for example, thiacloprid and also fluopyradiferone. So can we use the recombinantly expressed P450s, CYP9Q3 or 2, for pesticide risk assessment as molecular tools to predict toxicity against pesticides or pesticide mixtures? So to do this, we first of all looked for a model substrate, so-called fluorescent probe substrates, which are easily, I would say, oxidized by CYP9Q3. We found such a substrate, the name is BFC, and with this substrate, we developed an assay. So this is the fluorescent probe substrate, and this is converted by CYP9Q3 into HFC, or a coumarin derivative, which shows fluorescence. And if you put this on a multi-well plate assay, and you can see here that with increasing concentrations of BSC, the fluorescence is increasing because the enzyme converts this compound here into HFC. If you are now adding a pesticide to this particular reaction, and the pesticide competes with the fluorescent substrate for the same site, for the same binding site, you would expect to observe a lower fluorescence signal. So an inhibition of the fluorescence, or so-called fluorescence depletion. And that is exactly what we observed when testing thiacloprid, acetamiprid, and imidacloprid. So the signal is not depleted by imidacloprid, but it is depleted by thiacloprid and acetamiprid, just proving that these insecticides are interfering with the same binding site as the fluorescent probe substrate. And this also correlates with the LD50. We also can use this system to address synergism issues of ecotoxicological concern. It is very well known that tank mixtures of insecticides, for example, in this case, thiacloprid and a fungicide, Prochloras, under field conditions seem to be or are in fact more toxic. And that is a paper published by the German JKI. And what we did a similar experiment under laboratory conditions, and we found out that thiacloprid toxicity is synergized by DMI fungicides, in this case propiconazole, as well as prochloras, as you can see here, because thiacloprid in combination with the two fungicides in lab assays turned out to be highly toxic. We observed very much the same with acetamiprid, showing a very good synergism in combination again with prochloras and propiconazole. So we used our assay to show that this is due to the inhibition of CYP9Q3 by these two fungicides. And as you can see here, both prochloras in red and propiconazole in orange inhibit this particular reaction. And they inhibit CYP9Q3, whereas another DMI fungicide, proteoconazole, is not inhibiting this reaction. It could mean that this particular compound could be mixed with thiacloprid under field conditions without harming bees or being synergistic. At the next step, we looked into the literature, and there was a paper published in 2004 by Iwasa, and they tested a number of azole fungicides in combination with acetamiprid. And what they observed was a high level of synergism with different azole fungicides, at that time claiming that it is due to P450 inhibition without knowing which P450 is inhibited. And we looked into this, and we found out by doing inhibitory studies with the CYP9Q3 and CYP9Q2, and we got a very nice correlation between the LD50 based on synergism and the IC50 value, the inhibition of the enzyme. So here, I think we can conclude that the observed synergism in this particular paper is due to the inhibition of CYP9Q3. There have been other papers in the past saying, okay, in this case, double trouble, tank mix of thiacloprid and EBI fungicide, ergosterol biosynthesis inhibiting fungicides, and they did a field study, and they showed that compounds like prochloras and propiconazole inhibit or are more toxic in combination with thiacloprid. And we looked into our assay and were able to show that CYP9Q3 inhibition is not observed for this azole fungicide. And we can claim in this particular case that there's a 1,000-fold difference between prochloras and this fungicide. So there's no double trouble with this particular fungicide, just based on the molecular assay we conducted. Synergistic effects have also been described with fluparadiferone, in this case, a paper by Tosi and Ni in Proceedings B, and they also showed that propiconazole affects the metabolism of fluparadiferone by increasing its toxicity when it's mixed together. We know from our biochemical experiments that propiconazole inhibits CYP9Q2 and CYP9Q3. So I think that is a clear observation that the synergistic effect is based on the inhibition of these two enzymes. So a paper published in Trends in Ecology and Evolution said, okay, why are we not using pharmaceutical or methods which are used to explore, for example, drug-drug interactions in the pharmaceutical industry? And that is exactly what we are doing here, because I think that our molecular approach aims in really to significantly improve the efficiency and sensitivity of pesticide research and evaluation in terms of pollinator safety. So this particular investigation has been submitted for publication as a complementary approach for pesticide risk assessment at the molecular level. But what about bee pollinator toxicogenomics beyond Western honeybees? We also published papers that we that, for example, orthologs of CYP9Q3 are present in the genome of Osmia bicornis, a solitary wild bee species, a red mason bee. And so we sequenced the genome, and we found these two P450s, which also detoxify thiacloprid, similar to the buff-tailed bumblebee P450s, which are named 9Q4, 5, and 6, also showing detoxification of thiacloprid. So is it an evolutionary conserved mechanism we identified here which provide selectivity to thiacloprid and other insecticides. But we also looked into other bee species because we first checked managed bee pollinators. One of the major managed bee pollinators for alfalfa seed production in the US is Megachile rotundata. And we looked into the P450 phylogeny, and what we observed that a CYP9Q-like branch is empty, and they do not have an ortholog for CYP9Q we found in honeybees, red mason bees, and bumblebees. And that's what you see here, and that has been published by the end of last year in Nature Ecology and Evolution. So we found out that the leafcutter bee is much more sensitive to thiacloprid, acetamiprid, as well as butenolide insecticide than other managed bee pollinators. This knowledge can of course be used in order to mitigate the risks for these particular bee pollinator. Just to show you, if you apply these compounds, imidacloprid is highly toxic to honeybees. That's what we know in acute toxicity tests. Thiacloprid, taufluvalinate is a compound used to control varroa mites using in-hive applications. So it's of course also practically non-toxic to honeybees. The same is true for fluopyradiferone. But Megachile rotundata lacking CYP9Q orthologs shows high sensitivity to all these compounds across different chemical classes. And I think it is something very important also to investigate with other bee species. Therefore, we thought it is not the academia or the colleagues out there which are, of course, showing growing interest in other bee species for pesticide risk assessment. For example, stingless bees from Latin America. 20,000 species, I think that is very difficult. We cannot provide a resistance, a pesticide risk assessment on an in vivo level for so many species. It is impossible. So other approaches are necessary to do so. And what we did, we looked into the public gene sets and found gene sets for 64 different unique species. And so we also curated 9 CYP9Q-like genes from different bee species. And so this is a list we assembled, and all these P450, all these orthologs of CYP9Q3 from different bee species have been synthesized and functionally expressed in hive cells. In fact, we expressed more than 20 orthologs of CYP9Q3 coming from 12 different bee species out of 6 Apoidea families. And you can see here the families Halictidae, Collettidae, Andrenidae, Melitidae, as well as Megachilidae. We used our fluorescence depletion assay we developed with this model substrate to show that, or to explore whether thiacloprid also with its orthologous genes competes at the active site. And that is here for 6 different bee species. And what you can see here, that the orthologs from diverse families of CYP9Q3 also seem to bind thiacloprid in their active site. And that is a very promising result, I think. And to make it a little clearer, so we have the family of Apidae with more than 5,700 species. We have the Collettidae family with 2,000 species. And we have the Halictidae species with more than 2,000 species. But we have identified in all of these different families orthologous genes which are able to detoxify thiacloprid. And that could mean if we take this phylogenomic tracking approach, I call this phylogenomic tracking of conserved detoxification principle. Yeah, and that is something we can define nodes in the phylogenetic tree which help us to say, okay, everything what's below this particular node has an orthologous gene which is principally capable of detoxifying a particular compound. This is the approach we are currently following. And here we are working together also with other groups. And so, of course, we can also not express CYP9Q orthologs of 10,000 different bee species. That's also not possible. Now we have to take another approach, an in silico approach by computational modeling. We need a computational method in order to identify signature amino acids just by sequencing. And if these particular signature amino acids are present in a P450, it could mean and explain selectivity. We also did homology studies and molecular docking studies, which help us to explain and help us to identify the signature amino acid in the active site of honeybee P450s. Here we are collaborating with the Center of Computational Toxicology and Exposure with Carly Leloon from EPA, because they developed an approach which is called SECAPES, sequence alignment to predict across species susceptibility. This tool is currently tested with our in vitro result, because the testing of this tool was hampered by the fact that there's no data out there in order to feed it into the system. Now they have the data, and we are collaborating here with Carly Leloon in order to check if we can find a certain pattern, or if we can really identify signature amino acid, helping us to find out and fish in silico for orthologs, which may provide selectivity to certain insecticides. That said, the story continues. There's special interest also expressed in Akali bee, which is a very important pollinator in the US, also for alfalfa seed production, and it's nesting in salty alkaline soil. And the genome has been recently sequenced. It's public on NCBI. We curated the SciPome, identified 42 different P450 genes, identified a candidate CYP9Q3 ortholog, which has been cloned, expressed, and we are currently functionally validating it for its ability to detoxify certain insecticides. That said, I'd like to briefly summarize our novel approach. There have been a number of papers in the past by, for example, May Birnbaum on the xenobiotic detoxification pathways in honeybees. That's some further reading, also by Christina Grosinger on the power and promise of applying genomics to honeybee health, but also this particular paper asking why are we not using more molecular methods in pesticide risk assessment to have a higher throughput. And I think our approach on bee toxicogenomics and phylogenomics helps to understand and explain issues of regulatory concern. Just remember the mixture studies we did. The mechanistic studies help to identify and understand evolutionary conserved molecular principles of selectivity among bee pollinators. It helps us to track the selectivity and identify conserved detoxification gene families, and may help also to cluster tribe-linked sensitivity issues, as I've shown you with the nodes in the phylogenetic tree. Biochemical high throughput screening assays are important because based on those P450, they can help to predict speed and extent of metabolic clearance of candidate insecticides, which are, for example, in early discovery. We can use these particular methods in order to just sort out certain chemistry which are not detoxified, and just following those which are detoxified by these particular enzymes. And of course, we'd like to strive for science-based communication with the regulatory authorities worldwide, because they'd like to understand. It is no longer enough to tell them that an insecticide is not toxic. They'd like to know why is it not toxic. And if you can explain it by molecular means, I think that is very important. Last but not least, I have to thank my team, which is dedicated to do this work, and special thanks to my lab, what you can see here, and a number of PhD students are currently in my lab. Julian did a lot of this phylogenomics work, and the expression work was done by Bettina, and we have Deborah is a postdoc working in my team in the bee toxicogenomics project, and also former students such as Marion or Catherine Beadle, Jillian as a postdoc, Christina. They did a great job in helping us to understand the principles underlying insecticide selectivity in bee pollinators. And last but not least, my long-term collaborator, Chris Buss at Exeter University, and some of his team members. I'd like to thank you for your attention and I'm happy to discuss with you later this particular presentation. Thank you.
Thanks, Ralf. That was very interesting. I think we have some time for questions. Is that correct, Natalie?
Sorry, yes. Yes, we do. If there are some questions from people who are not in the class, we should definitely prioritize them. Thank you.
Sure. Feel free to enter questions into the chat or into the Q&A if you have any for Ralf.
It looks like there's a couple of questions in there right now from Christina. Can you see them, Ralf, too?
I'm sorry?
Can you see the questions in the Q&A?
Yes. Let me see. Oh, yes. Okay. So the first question is on the fluorescence assay. Much more high throughput, faster than doing assays with live bees. How many pesticide functions can you screen at one time? Yes. So that is, I think it's indeed very fast. And we can test hundreds or even thousands of compounds a week. I would say we are doing this in 384 well plates, and similar to what the pharma industry is doing on CYP3A4 testing with what is a human P450 involved in drug-drug interaction, possibly involved. And so we can indeed test thousands of compounds even a day if we want. So that's no problem. But at the end of it, we also found chemistry which is not detoxified by CYP9Q3. That is very important. So that's it. Not all insecticides which are selective are detoxified by CYP9Q3, but we may identify different P450s which can be used in this particular assay. We know that chlorpyrifos and taufluvalinate are detoxified. That is work from May Birnbaum and Wenfu Mao, and we know it also from a number of compounds we tested. So, and the other question is there variation among nicotinic acetylcholine receptors targeting chemicals in terms of their concentrations in plant tissues? Are there some that are less likely to concentrate in pollen nectar versus leaf tissue? Of course, I think it depends on the physical chemical properties of the insecticides you are considering. Those which show a fairly good systemicity tend to show a higher distribution in the plant than those which have a higher log p value and are less translocated. But it is also something you can really, I would say, change by modifying the chemistry, and that is something. I'm not a real chemist doing discovery and insecticide synthesis, but I think that is something which can be you can overcome this. Yes, there's another question. If we have considered widening our approach to understand toxicity of neonics against predatory insect species. Yes, we thought about this, but I would say my resources are not endless, and that is the main point. I would love to, for example, to also look into other organisms, hymenopterans in particular, such as wasps and ants, in order to look if if they also carry CYP9Q orthologous genes, in order to see how far this particular detoxification principle is conserved in evolution. given all these uncertainties with testing pesticide effects and their combinations, do you think there should be more effort going into researching non-chemical crop protection methods? I think it is something which is addressed anyway. I'm an insect toxicologist working in research for my entire life, I have to say, and I'm normally not heavily involved in any marketing aspects of these of these issues. So that's of course, I think there are non-chemical crop protection methods which are useful. Say for example beneficial insects. I think the entire seminar is on integrated pollinator and pest management. And integrated pest management of course includes biologics. And I think our studies may help at the end of the day to better understand also possible interactions between, not only between pollinators, pests, insecticides, but also biological control methods you can use in order to control certain pests in combination with insecticides.
So in the interest of time, because I know we do have another follow-up student discussion coming, I think we can handle the remaining question in our class discussion, if that's okay with all of you.
Okay.
Okay, so I think we can conclude the seminar for now. Thank you so much, everybody who's been able to participate, and we're going to take some of the students with us now and enter a private discussion. But I appreciate all of your interest and questions, and we'll catch up with you again next week. Thank you.
Thank you. Bye-bye.
Thank you.